I want to thank the organizers for the opportunity to participate in this completely delightful meeting today. For those of you who've been asking me, and this is going to be my topic today, I changed the title just a tiny bit. And if you're interested in having the answer to the clinical side of this particular question, you should go see poster number 26, because I'm going to talk more from the basic side. Many people have been asking me, why I actually recently moved from New York to Tucson. So this is as I would walk up to my building in the Bronx in February. This is what my walk up to my building looks like now in February. So I don't think I have to say anything else. Kind of clear, right? So this is how, so for those of you who heard my talk at the mini course, you can go to sleep for a couple of minutes. I usually start my talks the same way. So from my standpoint, part of, and I've been working in this subject for about 20 years now, frightening though it is to say. Part of the problem, I think, with studying hypertrophic cardiomyopathy from almost any standpoint is not only the variability in the disease, but the variability in how we look at the disease. I just want to point out before anyone gets profoundly insulted, I've actually worn each one of these 4 hats. I'm a practicing cardiologist. I started my career as a geneticist. I'm sort of a junior biophysicist at this point, thanks to people like Corrado. And I'm, of course, as all internal medicine people, I am still an internist. So the question becomes, when you ask each one of these groups what this disease is, you're going to get a very different answer. So any cardiologist will probably talk about, as we say, massive hypertrophy, sudden cardiac death, chest pain, obstructive disease, of course, diastolic dysfunction. I always like to point out at this juncture that this diastolic dysfunction is often out of proportion to the degree of hypertrophy. Barry Marin pointed this out many, many, many years ago. And to many of us sort of speaks to the fact that this diastolic dysfunction has a cellular basis. But as you can see, it's a wide range of opinions on what the disease actually is. My geneticist colleagues, well, this is my family, you can't have it. There are, an endless array of causative mutations. There are no genotype-phenotype correlations are possible because the degrees of freedom are just way too wide. So, my usual view of that is something like this. I know there are 1,000 mutations. I think we can do better than just sort of throwing our hands up. From the biophysicist side, we're really not a whole lot better at this, where we all pick our favorite protein. That's the center of the universe, of course, and the rest of the proteins in the sarcomere, which is truly a giant machine, are kind of unimportant. And of course, I have the very best assay, and that's your answer. You can see sort of my frustration here. And of course, the poor internist who's like, this is a pretty common disorder overall, and if all of you would stop arguing with each other, maybe we could get an answer on how to manage it. So I've long referred to this as sort of 6 blind fields in the HCM elephant, and this is, of course, from the old parable of the 6 blind men who are holding on to different parts of this elephant and describing something completely different. Well, that's really the state of the disease at present. I want to argue that 20 years after the linkage analysis, and I can tell you from having been around back then, that we really thought, much to my embarrassment at this moment, that this was going to be, excuse the term, a slam dunk. Let's think about this. These are proteins where we knew exactly, to a large degree, what they do, where they are, how they function, and we thought we'd explain this really quickly, and we have not. And of course, the key being to develop these genotype-phenotype relationships such that we can really have genotype-driven management and eventually real therapeutics. And I think this dovetails really nicely with what Jacopo was saying a few moments ago, where we need tailored therapeutics, and I think the genotype is going to be part of that, but we need to do better. Just some basic clinical observations. I'm certainly not going to go into anything like the folks in the last session. Again, this diastolic dysfunction is out of proportion to the degree of ventricular hypertrophy. Frequency of sudden cardiac death does not track necessarily with LV size. Yes, in the extreme cases of greater than 30 millimeter septums, yes, but it doesn't necessarily, a small ventricle does not mean that you have a low risk. Preclinical HCM caused by sarcomeric mutations is characterized by impaired relaxation but preserved strain. Carolyn Ho has shown this very nicely. And recent longitudinal follow-ups of genotyped cohorts now, and again, take a look at poster 26, suggests that the natural history of this remodeling disorder is progressive in many cases. This is exactly what Jacopo was saying before, and I really want to emphasize this because this is the way I think of this, as a remodeling disorder. How much remodeling you get is to some degree driven by the mutation that the patient has. And we know that sarcomeric mutations now cause both hypertrophic and dilated remodeling. I want to point out, I'm going to talk about the cardiac thin filament today. I want to point out, interestingly enough, that TNT, I think at least 20% of the known mutations in TNT actually cause a dilated disease. So this is a very, very complex sort of biophysical mechanism that is driving this remodeling. I just want to point out that we've actually had some good experience in phenocopying these models, and we'll talk about why this is important. I just want to point out, so here's a TNT mutation that's a single amino acid in frame deletion, normally quite severe mutation in patients, and all I really want you to look at, these are mouse hearts at different ages where we've expressed varying degrees of the mutant protein in the heart, either getting a 35% or a 70% replacement. All I really want you to see is that this is a normal sort of progression in a normal mouse from 6 to 12 months. You can see in these animals as early as 6 months, and we can detect this actually at about 3 months, there's severe remodeling of this ventricle. This pointiness is very, very reproducible. Say 12 months later, it looks like a tiny baseball or some sort of ball as it rounds up. And if you look at this 12-month and the higher expressing at 6 months, those two are pretty much identical. The point I'm making here is that the amount of protein seems to accelerate, if you will, the actual development of the end phenotype. And I think this is very important. But this is the sort of progression, as you can see here. We call this one of our train wreck mice. You can see that the remodeling can become very aggressive and very severe, and this animal will eventually dilate. You can see early bits of this here. So I just mentioned that you can also have dilated cardiomyopathies in thin filament proteins. And I'm showing this actually for just about the first time. I just want to say this is the work of Lauren Tall in my lab, an MD PhD student. And what she did was she took a tropomyosin mutation that was extensively reported on, again, by Carolyn Ho and Elizabeth McNally, that had a very interesting bimodal phenotype. I won't talk about that today, but I just want to show you that just expressing as little as 10% in this column or 50% of the abnormal tropomyosin causes what is a very clear dilatation here. As early as 2 months, we can pick it up in a high, being just 50% transgene protein. If you follow this column down, you can really see large chamber, very thinned walls. It just continues, less so in the lower expressing line, but still a considerable dilatation that is progressive and primary. Okay, there's very little fibrosis early on in these mice. This is a structural cause of dilatation. So how do you integrate all of this? And this is sort of the point I really want to make, is that, and this is my sort of joke about us biophysicists who take one assay and just run it into the ground. That really basically doesn't work, because we've had to look at this entire problem at multiple levels of experimental resolution, if you will. You cannot do it just with one approach. I've learned this, trust me, the hard way. I've started as a cardiologist in physiology building murine models, and I think they're basically essential for studying, of course, a progressive ventricular remodeling disease, where we've been able to measure calcium kinetics, energetics, LV remodeling, things like heart rate variability. We can do all of these in the mice. About 6 years ago, I became a little bit dissatisfied with the mechanistic insight I was getting out of this and moved back and started looking at some of these biophysical approaches. The key to doing this is actually to couple these approaches, not just to sort of randomly take one assay and try to correlate it, but to very carefully match these. More recently, along with Steve Schwartz, also now at Arizona, we started to look at computational models, and this was actually we've been doing this about 5-6 years, where we started with protein fragments, and we've worked up to atomistic and now coarse grain models of the entire thin filament, and used this as a laboratory to integrate with, in particular, the regulated in vitro motility assay, to give us a continuum, if you will. Of course, the last aspect to this, of course, is to add patients and to truly understand the pathogenesis of, again, this very complex form of the disease. And so in comparison, I don't know if you were here for Jacopo's talk, and he was sort of talking about the two of us, bantering over echoes. Out of that bantering session grew this consortium from the Brigham, University of Michigan, Stanford, Arizona, now in Florence, where we're actually looking at these HCM patients, and we're using clinical observations and going back into our computational models, back into our murine models, and trying to build true networks that explain the mutation-specific remodeling that we see. This is quite frankly, from my perspective, has been very fruitful, I think, and is really the way to get sort of global answers. One way just doesn't work. So here's the thin filament, just to remind everyone. This is actin. This is actually the atomistic model. Tropomyosin here, helical coiled molecule, lends strength to the thin filament, overlaps in the N and C termini. In yellow here is TNT, the protein I'll be talking about mostly today. Blue, TNI, and red TNC, the calcium sensor of the thin filament. This is a machine, a highly regulated, allosterically regulated machine that determines the calcium concentration in the cell, can be phosphorylated at multiple sites, and modulates myofilament activation. It modulates how the sarcomere works. Okay? So the bottom line is, when you disrupt that machine, you get disease, and that's the question we want to answer. This is an old slide. This is actually it's a slide that's actually readable. There are well over 40 mutations in TNT now. I'm going to focus down here in this part of TNT for which we have not a ton of structural information. I'm going to look particularly today at the R92 hotspot region. And this has actually been described for many years. Patients with each one of these mutations at R92, Q, L, or W, have all been isolated in patients. The patients are different. My mice are different. So it's a very nice system to study. So let's start with a clinical observation. So this has actually been seen forever, that HCM patients seem to have this blunted beta-adrenergic response. Below here is really sort of my rubric or my paradigm for how I think the disease works. And that's that we have these primary myofilament abnormalities that are caused by these mutations that lead to changes in mechanics. You can have isoform swaps. You can have major changes in energetics. This is part of, I don't know if you noticed, but Cecilia a moment ago sort of mentioned this issue of ATP utilization. Corrado Pugliese mentioned this the other day. This is very, very important. We've shown this at the animal level. It's also been seen in patients. I just want to reiterate that this is a biophysical disease. This is where the disease starts. There's a lot of variability in this disease, but every patient with a given mutation should have the same biophysics, and we need to focus on that to some degree. The degree of these changes leads to, of course, pathological remodeling. There is a second axis that we really cannot ignore, and that is the calcium regulatory axis. We don't understand 100% why, when you alter thin filament mutation proteins in particular, you have a downstream effect on calcium handling, but you do. It's a very early finding in some animal models, certainly, and it's a vast contributor to this overall milieu. So you can well imagine that if you have a mutation that greatly alters mechanics and doesn't affect calcium very much, you may have one pathway for remodeling, while if you have a mutation that hits the mechanics and the energetics and calcium, you'll have it different. And this is exactly what our animal models have shown. I want to emphasize this because I think this is going to be the basis for how we start subdividing these clinical cohorts based on genotype. And of course, these relative contributions develop the severity and the time course. Again, progression. How fast does it occur? How inexorable is it? What can we really do? So this is just a simple description here. So here we have, if any of you have really good eyes, you can see that these tracings look really old. This is from the R92L mouse. We've been studying this mouse for almost 10 years. All I want you to take home from this is just a simple intracardiac tracing, pressure tracing. Here, baseline up is contractility, or the positive dPdt. Down is negative dPdt. The addition of a beta-adrenergic agonist, dobutamine, predicted increase in the rate and the amplitude here of the response. Contractility and relaxation both have to speed up to maintain an increase in cardiac output. The R92L mouse, which is right down here, and I'll mention this in a moment, actually at baseline looks relatively normal. There's a little bit of difference, but you can see that this response to dobutamine is greatly blunted, particularly in the diastolic phase. Something I want to point out down here, so here are this is a 4-month panel from mice. This is R92W mouse, this is R92L mouse. The only difference between these two mice, they both have 50% transgene. The only difference between these two is that amino acid. Okay, so you don't have to be a mouse cardiologist to see how different these look. And if you have really good eyes, you can see that you get the atria and the RL are quite large. It's a profound finding. Calcium sensitivity of force generation, another parameter that we look at, many people look at the fiber level, does not determine this change in either function or in geometry. They are superimposable. All 3 of the R92 mutations have the exact same effect on calcium sensitivity of thin filament activation. It is not the determinant. And I'm going the wrong direction. There we go. This is just, I just want to point out, this is a compilation. These are the sorts of slides I show, and I cringe for my students because this is 3 or 4 students' PhD projects all on one slide, because these projects take years. I just want to point out that here are all 3 of the animals. These are 3 of the animals we've studied, yet another in comparison. And R92L, so this is the one with the blunted beta-adrenergic response, has always been a bit of a mystery to us. Those hearts don't particularly look very normal. And yet we don't see big changes in calcium homeostasis. The energetics via P31 NMR that we did with Joanne Amwell at Harvard, not really affected. We really didn't rescue very much with a beta myosin rescue mouse. We've just published this. And sarcomeric structure looks pretty good. But this mouse isn't normal. So Pia Ginto in the lab a number of years ago was studying the phosphorylation patterns. So part of understanding how the calcium handling was abnormal in many of these mice was, of course, to look at phosphorylation of, in this case, TNI, which, of course, is phosphorylated and leads to a decrease in calcium sensitivity and speeds relaxations, part of the physiologic response. Here's phospholambam, which is also part of the pKa axis we use as a control. The only thing I want you to take home from this is a normal mouse here, 1 and 1. And the addition here of pKa increases phosphorylation as you would expect it to. Mice are hyperphosphorylated at baseline, bear this in mind. PLB, the exact normal response. You expect to see this increase in phosphorylation with pKa. This was all done in hearts that were removed from the animal, perfused, and perfused with isopaternyl. What we found with R92L was quite shocking. I must have made that poor student repeat this about 10 times. You can see I do feel guilty. But in any case, what we noticed immediately was that the level of phosphorylation was very low. And this is TNI, right? The mutation is in TNT. Okay, so this isn't particularly obvious. Why would this be, right? So when we added isopaternyl, we saw a bit of an uptick, but if you look down here and you can see this shows the TNI phosphorylation. That uptick was not very substantial. We really couldn't phosphorylate this TNI in the context of this TNT. So of course, because I never failed to torment students, we insisted that she look at yet another mouse and repeat all of these experiments with yet another mouse to see whether this was some sort of general phenomenon. Then you'll hear me argue repeatedly that there are very few general phenomenon, I think, in this disease. Sure enough, when she looked at a delta 160E mutation, that was that other one I mentioned before, which actually happens to be located much physically closer to TNI and TNC, she saw none of this. Completely normal phosphorylation pattern, except for PLB, which I won't go into because there's calcium handling problems in this heart. But at least with regards to TNI phosphorylation, virtually the same. Okay? So this is specific to the R92L, that when you have this mutation, you cannot normally phosphorylate TNI either at baseline or in the context of beta-adrenergic stimulation. So there's no obvious answer to this, trust me. So our hypothesis was that we felt that there was an allosteric effect. Okay, so that the effect of this mutation far away, way at the other end of this molecule, down here, here's R92L, was able to be transmitted through the machine allosterically and affect phosphorylation potential at serine 2324, in this case with a mouse. The point I want to make to you is that that seems a little far-fetched, and it's certainly not the kind of hypothesis one would come up with out of the air. Because I'd be laughed right out of my entire career. But, you know, what became very clear was that we didn't, beyond the fact we didn't have any other ideas, you know, this is hardwired into the system because what we do know, of course, is that when you bind calcium in this portion of the molecule, of course, that is transmitted to the tropomyosin binding domain down here. So this is hardwired, this crosstalker, you will, or this propagation of these signals through these protein-protein interactions. So probably it wasn't such a great surprise. We were surprised, though, to see it so dramatically. Beta-adrenergic activation in the cardiac myocyte. So where is this uncoupling? It could be at multiple levels. Of course, you can have a change in the beta receptor density. We checked this. There was none. Beta-adrenergic receptor density is exactly the same. We could look. We already looked at pKa of TNI and phospholambam. So these were the things we were interested in. I had already shown that actually it was completely maintained. Remember, the phosphorylation of phospholambam was normal in these cells. So that wasn't the problem. The problem was at the level of the myofilament. So how do you test that? Well, so what we did was we obtained a phosphomimitic. So this is a mouse made by Anne Murphy at Johns Hopkins where the sites of normal phosphorylation in TNI have been replaced by aspartates, mimicking the charge distribution at that site. We call these constitutively phosphorylated, and these animals actually show many of the properties of a phosphorylated TNI. So of course that's the test. If we put this mouse, if we cross these two mice and we abrogate, if you will, or we eliminate the need for this TNI to be phosphorylated, we should recover function. So that was the goal with these studies. These took years. I just want to quickly point out that we were able to restore sarcomeric. I'll just show you different parts of this. We were able to completely restore the sarcomeric mechanics. You can see here in in green is the abnormal mouse. So this is the R92L. When we added the DD background, we received a much more normal pattern of contraction and relaxation in the unloaded cellular. Very excitingly to us, remember I told you when you muck about, with the thin filament, you can change calcium handling. What we found, and I think I did tell you that R92L calcium handling was basically largely normal. We were very happy to see that the double transgenic, which is here in the red, wasn't appreciably different with regards to the calcium kinetics. So it was whatever effect we're going to see in the final experiment that I'll show you in a moment was not due to calcium. Again, it was due to the myofilament. And here are sort of the results where we were able to rescue contractile reserve. Again, red is the abnormal mouse, and green here is the double transgenic. You can see with regards to an increasing dose of dobutamine, and again, here's positive DPDT. You see that the greens recovered most of their ability to respond, and probably most importantly is this diastolic effect from every single point was improved by this. Okay, so we rescued the phenotype just by adding in this TNT. This TNI with the double phosphorylation. One thing I want to point out is that the R delta mouse, of course, like I told you, never miss a chance to torture a student, made her do all of these experiments again in a delta 160E mouse, and we did not rescue as predicted because that wasn't the problem in those mice, as I showed you. It was not a myofilament error. So what's the biophysical basis of this? This is a really hard question to answer, right? How would you even begin to answer that question? Luckily for us, and so I just want to remind you, the mutation is down here, TNC and the phosphorylation sites are down here. Delta is closer to both of them, doesn't have this effect. So we turned to Steve Schwartz and Ed Manning and started to look using our all-atom model to see whether we could predict these changes in sarcomeric dynamics and whether we could see this propagation at the atom level. I'm not actually going to talk about this. This is all published. We first looked to see whether these effects in fragments that were used in the biophysical studies could show differences in the mutations. So this is just classical molecular dynamics, looking at force being equal to the negative derivative of potential energy. All you're doing with molecular dynamics is that you're looking at trajectories. You're looking at where the atoms go over time. Okay, so it's a great way to look at dynamics. It's not the way you define structure. Let me just add this. This is all published. I'm just going to point out we found mutation-specific effects locally. Something we found that was very shocking was that the effects at the mutation site were actually propagated through the helix. So this is an important point. It's been now seen in by multiple groups that when you have a helix, think about it like a slinky, if you will. If you hold down both sides of a helix, if you basically constrain it, if you will, and you change the forces at one part, that region has to, if you close it here, you have to open it here. Okay, it's basically potential energy. So this is what we found. This is very important functionally because remember I told you these are at a distance. So here we saw it at a very local distance. Why is this important? It's important because when you interpret these functional assays, let's say, and you've put the mutation, let's say, at residue 92 and you see an effect, you're going to assume that it's due to the change at residue 92, right? That's obvious. But in this case, it really wasn't true. The effect happened further down in the molecule, and what we think it actually did was disrupting this overlap, okay? Because all of the mutations that we looked at in this region changed the structure of TNT in the overlap. So we think that's sort of a theme here. I'm not going to go into this in any more detail. So sort of to go finish this off, we then went to our full-scale model, and this is a 20,000 atom model. Running the simulations for only, for example, a nanosecond take almost 2 weeks in our large supercomputer. And basically, we can put every last one of those, we can track the position of every last one of those atoms over time. So it's a very powerful tool because it allows us to look at these far field effects. This is the question we want to answer. And so this is just the sort of thing you can do with this. I'm not going to go into great detail, but I just do really want to point out how powerful we found this. So here we're just looking at TNT. So what we can do is we can put a mutation into the entire model, and then we can look at each protein and trace the trajectories of each atom in each protein to decide basically what's happening. This just gives you an idea. When we add or take away calcium, you can see these big fluctuations, and what this root mean squared fluctuation only means is it's a measure, in this case, of flexibility. Are the atoms moving far, basically, or a good distance in creating flexibility? And we recreated many of the findings that have been seen previously with regards to the flexibility of the molecule in these two regions. This is, by the way, the delta region that I'm so excited about all the time. And this is actually, this fluctuation here is probably why the entire complex could not be fully crystallized. And this part had to be cut off from the crystal. Sort of interesting. So what did we find? What we found was pretty shocking. So when I was doing these experiments, at the same time, the Schwartz group was querying, if you will, all the different many of the different mutations. We've covered quite a large number of them by now. And so I asked them if they had seen anything down in TNI, TNC. Was there any difference in that region? They frankly thought I'd sort of lost my mind because it was not something I usually asked them. So they said, well, we'll take a look. And what did they find? Quite interestingly, so here are all the known mutations in that 160 to 163 region. All this is a graph of the distance between the serines, the region of the serines on TNI, and the second site where calcium binds. So this is where pKa has to get in, right? It's the site where it has to come in to phosphorylate. For these delta mutations that I showed you had no effect. These actual distances were the same. There was no change really from wild type, which is over here. Not so for RL, where the intra-distance between TNI and TNC closed down considerably, all the way from around 15 down to about 7. So we think this is why pKa can't phosphorylate this region. It's a biophysical region. This is why the effects of the blunted beta-adrenergic activity are seen in this molecule. They can't get in. It can't be phosphorylated. Not quite the same for RW, which actually goes along with other data that I won't show you. And here's the map with the wild type, and here is the mutant one coming in very close. So I've actually already mentioned most of these conclusions. Of course, these beta-adrenergic responsiveness, this specific finding obviously has impact on how we might treat or approach these questions. We can use the molecular dynamics now to query all mutations and look at different effects on protein-protein interactions. And now what we're doing is validating these findings via FRET and other intermolecular approaches. I'm actually not going to talk about this today. I'm going to go ahead. But we have started to reverse the disease based on the calcium work. And these are actually the folks who do the work, certainly not me. This is Ed Manning, who did a lot of the modeling, along with Pia Ginto, Jesus Jimenez, who had to make endless numbers of mice, and Lauren Tall, who did the dilated mouse. And I'm going to stop there.
So very nice talk. Thank you very much. I wanted to ask you a question about the double transgenic. Mm-hmm. So you showed us nicely that you can rescue the function, but I saw that in isolated cardiac myocytes that there is a decreased diastolic sarcoma length, which was not rescued.
Correct.
So could you comment on this?
Absolutely, great question. So this is something we noticed as well instantly. And for many of the thin filament mutations, you know as well as I, there is this shift, the increase in sensitivity, right, the hypersensitivity. And again, I showed that at the fiber level for these. That, in many ways, I think is if you will, locked in. I mean, this is sort of the very, very dominant. And we were not able to fix that at all in any of the mutations where we've tried to do this. And it does seem to just be a dominant effect. And again, so the measurements were unloaded. And I think that's the trick. I think if we looked in a loaded system, we actually would see, so if we did fibers in these mice, I think we might see a bit of a recovery. But that's my thinking there.
And did you have the chance to measure diastolic function in these mice?
I'm sorry?
To measure diastolic function in these mice?
Yeah, in the rescue. Yeah, so they did rescue.
Are there any questions?
I have a question. So you mentioned, this is a clinical question. So you mentioned that this model has a blunted beta-adrenergic response. So how do you fit this with the beta blocker therapy that we give to the patient? So did you study the effect of beta blockers in these mice?
No, you know, the reason that we didn't, and we thought about it, and we're in the process now of testing, you know, many therapeutic agents on these animals. I think why I think it would not work, but I think that's actually an excellent experiment to show why it wouldn't work, is because the uncoupling is at the myofilament level. So my prediction there would be it wouldn't do anything, because in a way, when we stimulate with the dobutamine, obviously, we're showing the activation. So my guess is it wouldn't help, but it's a good test.
If I may ask you a quick question. There's an old paper, an old Japanese paper many years ago in patients, and this paper showed that patients with impaired contractile reserve with dobutamine, I'm not sure, with adrenergic stimulation, were more likely to evolve toward the end stage. So they had contractile dysfunction and dilatation, etc. And would this fit in your model?
Yeah, so that is an older paper, and that's a really interesting observation. Many of the Japanese groups have very, very interesting observations, and for the junior people, I would suggest you look back through some of these papers. Yeah, so exactly. So I pointed out, for example, the atria on these mice, which are simply enormous. So in my lab, we joke they have elephant ears. You can't be blinded when you study these mice. And they do progress quite a bit. And these animals, actually, what they really have is a very, very severe diastolic dysfunction that seems to get worse over time. And, you know, what the mechanism would be, why you can't, again, if we argue that the basic mutation causes this uncoupling at the myofilament level, why that would do this may have to do with the lack of ability of the heart to respond. So in many of these other cases, the other mutations we look at, for example, that have calcium misregulation, the heart clearly responds. That was the basis of the studies I didn't show you with CAM kinase inhibition. You know, the heart tries to fix what it feels as a calcium problem. These hearts, since they do not have a calcium problem, the heart really can't fix it. There's no way to really do that. And we think that that incapability of having any sort of compensatory response drives it down a much more progressive and more severe pathway. So it's a great observation. That's what we think.